Welcome to OA Week 2023. Thank you for joining us for this event. My name is Yuman, and I'm an OA Week coordinator. Please note that this session is being recorded. OA Week is presented by GOA-ON, the Global Ocean Acidification Observing Network. GOA-ON has three sponsoring organizations. First, the United States National Oceanic Atmospheric Administration. Second, the International Atomic Energy Agency, Ocean Acidification International Coordination Center. And third, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. GOA-ON was established in 2012 with just a handful of members. Since then, GOA-ON has grown immensely. It now has over 1,000 members from 114 countries. GOA-ON also consists of 9 regional hubs, which span across continents and oceanographic regions. We will be hearing from most of them throughout OA Week. If you are not a member yet, you can join GOA-ON today by visiting GOA-ON.org. OA Week debuted in 2020 and returned in 2021. When events and conferences were postponed due to COVID-19, following the successful in-person symposium on the ocean in a high CO2 world in 2022. GOA-ON is bringing back OA Week 2023 to maintain momentum around OA research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to present a wide range of ocean acidification topics and speakers from around the world. During the presentation, all participants are in listen-only mode. You're welcome to type any questions into the question box, which can be found at the bottom of the control panel on the right-hand side of your screen. We will be monitoring incoming questions and will pose them to our speakers during the question and answer session, which will begin immediately after the final presentation. For the discussion, you can also use the raise hand function in the toolbox at the bottom of your screen, and we will call on you to ask your question directly. And with this, I would like to introduce you to our moderator for this session, Azaria Pickering. Azaria Pickering is Technical Officer at the Pacific Community, working on their Pacific Community Center for Ocean Science. She graduated with a postgraduate diploma in marine science in 2022. Most of what she does is in the OA space, working with Pacific Island countries on OA monitoring programs, providing support and carrying out Fiji's OA monitoring program. Azaria, the floor is yours.
Thank you, Yuman. My name is Azaria, and I'll be moderating this session in the Pacific Islands and Territories Ocean Acidification Hub. And before I do, I'd just like to introduce this session. So increasing atmospheric CO2 levels and its implications on marine organisms raise the need for a multi-scale approach to ocean acidification research and monitoring. In this session, we look into processes and environmental factors that affect marine species in the context of ocean acidification, mitigation approaches, and monitoring activities in the Pacific region. And this session is hosted by the PI-TOA Regional Hub. So this morning, I'll be presenting on the Pacific Islands Ocean Acidification Center with a focus on, activities in the Pacific. Next slide, please. So ocean acidification most of us are familiar with the process or have in some way heard or learned about the process. And the other speakers will be going more in depth into the definition. So I'll just speak briefly on the chemistry side of it. So as atmospheric CO2 dissolves in the ocean, it alters its chemistry to form carbonic acid, and this dissociates into bicarbonate and hydrogen ions. And so the increasing supply of hydrogen ions limit the formation of calcium carbonate. And this is because the hydrogen bonds easily with carbonate ions rather than calcium ions. And so these chains of chemical reactions decrease the availability of calcium carbonate, reducing the growth rate of calcifying organisms such as mollusks, corals, and sea stars, etc. So why is this process important? Next slide, please. So ocean acidification has several impacts, and here I'll explain a few of them. The changes in ocean water chemistry can affect sensory systems, such as receptors that are used to detect chemical cues. And this reduces the ability of fish larvae to locate suitable habitats. One is the famous documented clownfish. Another impact is that it reduces the calcification rate of shell-building organisms like mollusks and crustaceans, and they become more vulnerable at larval stages. Corals also depend on calcium carbonate to build their skeleton, and so this process can slow down the building of their skeleton, leading to corals having a weaker skeleton. And the impacts of ocean acidification on fisheries and reef ecosystems ripple down to communities that depend on these resources for their livelihoods. For instance, us Pacific Islanders, we are heavily dependent on these resources. And so this is why it's important to monitor ocean acidification and its impacts. Which brings us to the establishment of the Pacific Islands Ocean Acidification Center. Next slide, please. So the center is a regional training hub that was established back in 2021 through partnership between members of SPC, the University of the South Pacific, NIWA, and University of Otago to support ocean acidification research and monitoring. And so the center provides support to Pacific Island researchers, government officials, and community members through training and technical assistance. And so while the awareness of ocean acidification is increasing, Pacific Island countries lack technical and human resources and institutional capacity to characterize ocean acidification. And so the center aims to address these needs and strengthen local capacity to respond to ocean change by providing hands-on technical training. For instance, we had a hands-on training earlier this year in February through the distribution of ocean acidification equipment that will help provide insights into local OA conditions, coaching and support with data management, and chemical observations to ensure that research meets global standards, access to international networks that support monitoring, action, and policy efforts, also local scholarships that enable cross-region collaboration and regional coordination across countries. So, sorry, next slide, please. What do we know in terms of what is happening in the region? So here you see an overview of what is happening in the regional ocean acidification space. And the map shows who is collecting data, what methods are used, what countries are using the the methods that different countries are using. And so we have well-established monitoring programs with some starting and some that have stopped but are still listed on the GOA-ON site. And it's important to note that this is a working map that was prepared by the center. And so we add information as it comes in. And so if you're not on the map, please do contact us. I think at the end of my presentation, I'll leave an email. And please feel free to reach out. Also, data has been collected as part of the PI-TOA network. And so there are a lot of people involved, different researchers and scientists. And so one of the challenges we found was when reaching out, because most of this work, it's done in silos. So one of our major challenges was gathering information from different actors to create this map. Next slide, please. Looking at OA capacity building in the region, with a focus on the GOA-ON in a box kit recipients, we look at countries in the region, what they are doing, whether it be their research plans, their monitoring programs, or future plans. And so from Papua New Guinea, we have Maisie, and she's the executive director for Fish Reef Project in PNG, and is currently enrolled at the University of PNG as a postgraduate. And so Maisie is among 5 other new GOA-ON in a box kit recipients, and she aims to use this resource to assess the vulnerability of nearshore and offshore reefs to ocean acidification through examining net community production and net community calcification. And she was also part of those that attended the hands-on training earlier this year, where one of the aim was to build capacity of scientists in the Pacific to monitor ocean acidification. Next slide, please. And from Samoa, we have Jacopo and Grady. Unfortunately, Jacopo has left us. He's moved on further, but we do have a new officer joining us from the Oceans Office in Samoa. And they're part of the Samoa Met Division. And in March, Samoa Met, in collaboration with Kiosk and SPREP, installed a OA monitoring buoy at Upolu. And so both of the recipients received training on collecting water samples, as well as the hands-on training this year. And so the team in Samoa are waiting to receive their kit, but for now they've been collecting water samples at the same site where they've deployed their buoy. And these samples are sent to Korea for analysis. And it's a collaboration between SPREP, Samoa Met, and Kiosk. We also have a second team in Samoa, and they sit at the National University of Samoa. Next slide, please. From Tuvalu, we have Po and Alitake, and they're with the Ministry of Environment. So these Alitake and Po took over Faulu, who was the initial kit recipient. And right now, Tuvalu is working with the government bodies to identify a suitable lab space. And this is also because Tuvalu has been facing challenges with using mercury chloride because they are party to the Minamata Convention, which permits them from using mercury. In hopes to address this, they'll be receiving a filtration kit along with other spare parts from the center. and they're looking forward to restart their monitoring program. They recently received support from the center and the Ocean Foundation, where we were able to facilitate the process of getting the ICME fixed and returned. And so right now they're planning to deploy the pH sensor after they've completed in-house testing. Next slide, please. And from Vanuatu, we have Tima and Marianne. They're looking after the kit right now, and through the support of Dr. Krishna Kotra, who was the initial kit recipient. And so they've successfully collected data for total alkalinity and pH data from 2019 to 2020. And they've also had capacity building workshops, which included university students to create awareness on ocean acidification. Unfortunately, due to concerns of management of mercury waste, the team had to analyze the samples the same day they were collected. So the center is going to be sending the filtration kits over. And so Vanuatu is waiting for that, along with some of the other instruments to replenish their kits. Next slide, please. From Fiji, so the kit was initially awarded to the Institute of Applied Science at USP back in 2018 to pilot the use of blue carbon on local mitigation of ocean acidification. And the data can be viewed on the SDG 14.3.1 data portal. So at the end of the hands-on training that took place in Feb this year, this marked the beginning of Fiji's ocean acidification monitoring program, where we're looking into understanding local ocean acidification at local level. And so every month water samples are collected and stored, and we have instruments like CTD and ICME that are deployed. Unfortunately, our ICME is not in water at the moment, but we're looking to prepare that and put it back into the water in the coming months. We've also done pH and total alkalinity analysis for 6 months, and we're looking into analyzing the next half of the year's water samples at the end of the year. And in progress of setting up the mercury filtration kit to filter out the wastewater. And in the future, we're looking to expand ocean acidification monitoring to include other sites around water level. Next slide, please. From Solomon Islands, we have Dr. Wycliffe and Dr. Linden, and they're also waiting for their equipment to arrive. They're one of the first new kit recipients to receive research stipend from the Ocean Foundation, which will be used for future training. And they've also involved students at the Solomon Islands National University in their monitoring program to build capacity, local capacity, and spread awareness on ocean acidification. Next slide, please. And so these are some of the country responses we got from some of the kit recipients following the hands-on training. And overall, the training was much needed, especially for those that are new. And the center, through collaboration with the Ocean Foundation, aims to continue this support for the sustainability of ocean acidification monitoring programs in the Pacific. Next slide, please. These are some of the challenges: lack of funding for ongoing monitoring, staff and equipment replacement, equipment breakdown, and difficulty in accessing or ordering spare parts. We have staff turnover, limited support for ongoing training needs, quality control and assurance, and a lack of collaborative support. Next slide, please. And looking forward, the center aims to seek funding for the sustainability of the center, expand monitoring and research activities, increase collaboration with other organizations, strengthen peer mentoring support, as well as increase monitoring support for Pacific Island countries and territories. And that's it. Thank you. I think I've gone way over time. But or maybe we could take a question or two. If not, then please feel free to put in your questions in the chat box and I'll respond to them as we go. But without further ado, I'm going to introduce our next speaker, Abby Smith. And Abby comes from New Zealand, from New England in USA, growing up on the coast of Maine. And early experiences involved wading and poking around tide pools, which sent her down a marine track. She majored in biology and geology at Colby College in Maine and spent the semester at the Bermuda Biological Station, where she developed a fondness for marine shells. And upon completing her master's degree at Woods Hole and MIT, she moved to Hamilton in New Zealand, where she completed her Doctor of Philosophy at Waikato. She's lectured and researched in the Department of Marine Science at the University of Otago for over 30 years. Alongside her work at the university, she has chaired the Otago Conservation Board, been a treasurer for International Bryozoology Association for more than 2 decades, and was the inaugural council chair for the New Zealand Ocean Acidification Community. She never lets her scientific career get in the way of her other hobbies, which include knitting, making jam, and enjoying cricket. Over to you, Abby.
Yoda, thank you very much. And I hope that I am now sharing. I don't think I'm sharing the right screen with you. Let me try that again. Good. Kia ora koutou, ko Abby Smith toko ingoa. My name is Abby Smith, and greetings to everyone. I'm here to talk to you a little bit from the point of view of being a marine biogeochemist. And ocean acidification is a perfect discipline for the confluence of marine chemistry, biology, and geology. And so it's obviously a place where I found myself after some years of not really knowing quite what I was. I look at shells and what they're made of, and how what they're made of has some influence on their fate. So if we dive into the ocean, biomineralization is when a shell takes chemicals out of seawater, when an organism takes chemicals out of seawater and makes some kind of skeleton. I mostly work on bryozoans, and this is a sort of a diagram of a bryozoan, but lots of different organisms do this. So we call this calcification, when they make a calcium carbonate skeleton. And then that animal dies, and the inorganic material settles to the seafloor. Maybe it breaks, maybe it whatever happens to it, it might get squished, it might get buried, and it might get chemically changed, or it might not. And at some stage, there might be uplift and some kind of fossilization, lithification and fossilization, and someone might discover a fossil. Unfortunately, all of these processes happen in a fairly non-random way, so that there are filters at some of these steps where not every organism calcifies, and not every organism that calcifies is buried and created into a rock, and not every rock is uplifted where we can see it. So the fossil record is really limited in what we can actually see. And we're really interested in biomineralization. Geologists like me are really interested in biomineralization because we can find some fossils, but we don't always know what they mean. Some of the things that affect biomineralization include things to do with the individual, like whether it makes different bits of its shell out of different things, whether it changes as it grows, whether if it has more than one shell, whether those shells are similar. There's also environmental things that affect biomineralization. Things like temperature and salinity and pH affect how things calcify, but also things associated with their ecology and associated with the fate of the shell after death. And then there's some of the characteristics of evolution that also affect biomineralization, possibly more than anything else. What you are or who you are tends to mean reflect what you're made of. So biomineralization is a very common thing. At least 55 phyla have some mineralized parts, and that's a useful thing, obviously, since so many organisms have adopted it. It provides protection, support, reproductive success, navigation, detoxification, and there are at least 60 known biominerals, most of which have calcium in them. The most common are bone, calcium phosphate, the thing that you're relying on right now to keep you upright, glass or silicate, which is what diatoms and radiolaria and sponges use, and shell or calcium carbonate, which is what I'm going to talk about now. The calcium carbonate is a very flexible mineral, and it comes in a lot of different, what you might call flavors, a lot of different ways to make calcium carbonate. The two most dominant in the sea, however, are calcite and aragonite. There are at least 7 polymorphs of calcium carbonate, but we'll talk about these two. Calcite is the more stable form. It's not so very soluble, and it's cheaper to produce, but it's not so very strong. And it can be, some of these little calcium triangles can be filled with magnesium sometimes, up to 22% magnesium. And the more magnesium you add, the more stress you put on the lattice structure, and the more soluble the material becomes. Aragonite, on the other hand, is not as stable at Earth's surface temperature and pressure, although it is almost stable. And it's more soluble in seawater, and more expensive to produce. So you wouldn't think any organism would make it, but it's much stronger than calcite, and is an especially good thing, and is more likely to be precipitated when magnesium is high in seawater. So when we look at, if we could imagine that skeletal carbonate mineralogy occupies a kind of space with aragonite on the left margin and calcite on the right margin, and various amounts of magnesium in the calcite, so up to 22% magnesium, with calcite, low magnesium calcite, intermediate magnesium calcite, high magnesium calcite, and very high magnesium calcite along that line, and how much calcite there is compared to aragonite along the x-axis, we can see that a lot of different organisms are clearly located in particular spaces. So eggshells and Codiacean green algae and corals and many bivalves are 100% aragonite. They live on this 0-0 axis. That's what they always make. They aren't very interesting. A lot of things are low magnesium calcite. They include the coccolithophores and many other plankton species, barnacles, crabs and other mollusks. And some things are very high magnesium calcite. These are really interesting, and people have done a lot of work on them. The red algae, coralline red algae, and echinoderms fall in that group. But then there's a group of organisms that seem to sort of occupy almost this whole space. And they include some mollusks, particularly abalone mussels, and bryozoans, which are my own particular area of interest. And I just want to congratulate Azaria for saying bryozoology so very well when it's a hard word to say. So I look at bryozoans. They are small colonial organisms. They have tentacles and a little bit of musculature, but they're not much to them. But they occupy these gorgeous little boxes, which they glue together to form large colonies. So each of these colonies is made of millions of these small boxes. Each of these boxes is no bigger than the point of a pin. They're tiny, and together those organisms work together to build these really complex structures. So even though the individuals are fairly straightforward, the structures are actually really interesting. Believe it or not, this is the most studied mineralogically invertebrate group. In almost no other context is it the most studied phylum, but it is in terms of minerals. We've measured the skeletal mineralogy of 5,290 specimens from all over the world, which encompasses 609 species from 132 families. So unlike many other groups, we really know a lot about the mineralogy of bryozoans. And this includes labs both in Aotearoa, New Zealand, in Europe, and in the United States that have done this kind of work. And so here's the data, unbelievable, gobs of spots. There are 569 spots here at 00, which you can't really see, and the mean is sitting over here. And what we see is that bryozoans occupy 62% of the biomineral space available to them. They don't make very high magnesium calcite, but they pretty much make every other combination possible. In fact, bryozoans are mostly all calcite, and then second mostly all aragonite, and then they have a variety of a few species that organize themselves in or specimens that organize themselves with both aragonite and calcite in them. If you look at weight percent magnesium, they certainly prefer intermediate magnesium calcite, but there's quite a lot of low and almost no magnesium calcite, but less in the high area. So the mean bryozoan is 82% calcite with 5% magnesium carbonate. But what's really interesting, I think, is that a lot of bryozoans combine calcite and aragonite. Again, I remind you, this is an organism that's essentially a little bag of epithelium with a couple of tentacles that can calcify. It's not a very complex organism at all, but they do quite a lot of stuff, and in terms of mineralogy, and it's interesting to look at that and see why. So this bimineral mineralogy has been reported in over 50 species. So if you look at just those specimens, you'll see they fall into three groups. This is the group that's mostly aragonite and has some high-ish magnesium calcite in it. This is a group that is 100% calcite, but it produces one mineral with not very much magnesium and one mineral with quite a lot of magnesium. And this is a group where they tend to make intermediate magnesium calcite, but they have an enormous range of possible amounts of aragonite and calcite. And I'm going to look at these kinds, an example of each of these kinds. This is the mostly aragonite kind. And if you look at along the length of a frond, distance from the growing tip here, you'll see that they high magnesium calcite, the weight percent magnesium calcite is highest in the youngest material and disappears as it grows older and older. The same thing basically happens with Melissaria chathamensis, this gorgeous bryozoan here, which if you stain it for magnesium, you can see that the lower magnesium content is at the tip. So this one, if you go the length of a branch, you will see that there's more high magnesium calcite with age. So here's the growing tip. The younger stuff is lower, and it gets higher and higher as it gets older. And this one is the most obvious one. This is Foveolaria cyclops, which makes an aragonite skeleton. And then as it gets older, a calcite skeleton. And as it gets older, it puts globs of frosting on the surface, which are aragonite. And you can see that quite clearly on here, that there's a base skeleton and then globs of material that has been added later. And this is stained here to make all the aragonite purple. So you can see that this organism is an organism that, like the other ones, makes a base layer and then thickens it secondarily as it gets older. So all of these are a kind of thing where there's a basic skeleton and then it adds more material as it gets as it grows. Although which minerals it's using varies. And why would they do that? Well, one reason is that there are different jobs for different minerals, and organisms that are biminerals sometimes make a bit of them out of a different thing. So you could think of a squid beak, which is different from the squid pen, for example. That doesn't apply well to bryozoans. Precursor, maybe making something that's easier to make and then using that layer to make something that's harder to make is a possibility in a chemistry solution. There's our sacrificial theory that that bimineral organisms might use a cheap outer layer to protect their inner layers. And there's a buttress idea where you add expensive outer strengthening once you get big, or an economic notion where you add expensive outer strengthening when you are big enough to be able to afford to spend the energy on it. Or there's an idea of a synergy that actually, if you have two minerals, you get the benefits of each of them, and they counteract the disadvantages of each of them. So this is important because carbonate mineralogy is one of the factors that influences dissolution rate. And I'm going to stop there and take questions because I have reached the end of my discussion. Thank you very much.
Thank you, Abby, for a very, very interesting presentation and taking us through biomineralization and skeletal examples of skeletal carbonate composition. I'm just looking at the question and answer box right now. We do have time. We do have 5 minutes actually for questions. If anyone has any questions for Abby. I'll probably give a minute or two. So
I did want to just share my screen to thank all the people who have done all that kind of research. These are some of the many people that have done this kind of work.
we do have a question for you, Abby.
Super.
So this is from Alexis, and she's asking, how can the Pacific Islands Ocean Acidification Center kit recipients study biomineralization processes?
Biomineralization is not biomineral processes are not very hard to study. You need X-ray diffractometry or some method of telling the difference between the different minerals, because they are actually calcium carbonate. They're the same thing, but they're different sort of arrangements of the chemistry. So certain machines simply won't test it, but X-ray diffractometry and XRF in using a scanning electron microscope are both ways to differentiate between minerals. An XRD, an X-ray diffractometer, is not a particularly expensive or difficult instrument to get at. University of Otago's one broke and then was not replaced, and so I actually send my specimens to Auckland, and they run them for about $40 an hour. It's not a very expensive thing to do, and then you get the data back. What you have to do though is it takes a little bit of experience to interpret what you've got back. The numbers are not always as clear, or what you get is a spectrum, and it's not that easy to interpret them. But once you practice a little bit, it's really quite an inexpensive and reasonable thing to do in terms of finding out more about what things are made of.
Thank you, Abby. Caitlin's asking, do you have a sense of how changes in ocean carbonate chemistry may alter the mineralogic choices of bryozoans?
Absolutely. And that was the second half of my talk if I hadn't have gone on and on. We did an experiment where we put bryozoans in different acidity, bryozoan skeletons in different acidity. So we weren't looking at living organisms, but just at what happened to the minerals as they sat there in different pH waters. And we had thought that the aragonite would dissolve faster and it would be very straightforward. And it turns out not to be straightforward at all. And mineralogy is certainly one of the characteristics that influences dissolution rate, but probably way more important is surface area and surface to volume ratio, surface area available to the fluid, and kind of overall architecture. So if something has got lots of little bits and crannies where the water can get in, it dissolves more rapidly than if it's a good solid block with not too much surface. And I don't think that the community has really understood the many facets of things that influence their dissolution history or their dissolution process as well as they could. And that's an area, I think, for chemists and geologists to work together more closely, but in order to tell the biologists what's going on there.
Thank you, Abby. We also have a question from, sorry, there's no name. But anyways, how do you sample your specimens before testing?
So the best thing is to collect something that's either alive or recently dead. Scrape away all of the organic material as best you can without doing any chemical treatment. So we tend not to bleach our samples, or if we have to, very, very lightly bleach them to clean them out. And then we take subsamples. Ideally, you take 3 or 4 subsamples from each part of the organism to make sure there isn't too much variation within the organism. And the sample could be about 1 gram or 2 grams of carbonate. And then we crush it into a tiny fine powder and put it in the XRD for analysis.
Thank you so much, Abby. We do have another minute to spare and welcome any questions that you have for Abby. Otherwise, I would like to take this time to introduce our next speaker.
Ngā nui. Thank you.
Thank you, Abby. Cliff Law. So Cliff is a principal scientist at NIWA, where he leads the Ocean Climate Interaction Group, and as a professor in the Department of Marine Science at the University of Otago. His expertise is in marine biochemistry with research interest in marine trace gas cycling, the controls of phytoplankton growth and impacts of ocean acidification and climate change. He has coordinated and collaborated in a number of international and research projects as well. And without further ado, I'll pass it on to Cliff.
Thanks, Azaria. I'll just share my slide and apologize for the fact that it's not in full presentation mode, but you should still be able to see it fairly clearly. So kia ora koutou floor aho. Today I'm going to be talking a little bit of a different subject from our discussions so far, and I'm going to be focusing on the role of the ocean in taking up atmospheric carbon dioxide and the potential to enhance that. And so this is a very topical issue at the moment. So I'm just going to be looking at a couple of the factors that we should be thinking about in anything we do around marine carbon dioxide removal. So this is just a schematic that I've put together of some of the techniques that are being considered out there in terms of marine CDR. So first off, let's establish what I mean by CDR. I'm referring to the fact that this is all anthropogenic activities that remove carbon dioxide from the atmosphere and durably store it. So the emphasis here is on things that we do, not natural processes in the ocean that take up CO2, but the things we do to enhance that uptake. And you can see in this figure here, moving from the left to the right, you can see probably things you're more familiar with in the ocean acidification field in terms of some of the remediation techniques around coastal blue carbon. So that's things like mangroves, seagrasses, and salt marshes. But then as we move to the open ocean on the right-hand side, there's other techniques that are being considered, such as the increase or enhancement of alkalinity of the ocean. There's macroalgal farms or cultivation. There's increasing the nutrient content of the surface ocean by artificial upwelling and deliberate release to stimulate phytoplankton growth. And on the far side, there's injection of liquid CO2 into the ocean. So these are just some of the techniques that are being considered. And so taking a step back here, these are getting more attention these days because of the state we're in of the climate, and particularly the fact that probably the Paris Agreement has given a bit of lever towards this. The fact the recognition is to maintain atmospheric CO2 and keep global temperatures below 1.5 degrees, we not only need to reduce our emissions, but we need to implement some sort of techniques to remove carbon dioxide from the atmosphere. Now, the ocean, as I'm sure you're aware, is an important sink for a lot of the CO2 that we've emitted. And so that is being considered as part of the solutions. And that's where these ideas come in. So as I said, there's many things we ought to think about before we do put any of these techniques into action. And I've been working with colleagues, international colleagues, and some of these are shown here, Phil Williamson and Naomi Vaughan from the University of East Anglia in the UK, and Leonard Bach, who's at the University of Tasmania down in Hobart. And we've been looking at different aspects of what you should really consider before you even think about marine CDR. And one of the really big topics or considerations is additionality. So what do I mean by additionality? What this is that ultimately for any MCDR, the acronym for marine carbon dioxide removal, any application of this should result in an outcome that's quantifiably distinct from the baseline that would occur in the absence of that action. So really the summary of that is that anything we should do should have a significant positive effect on the amount of carbon dioxide that's taken up. So that's an easy thing to say, but you need to consider quite a lot in this additionality. And so I'm going to give you a kind of breakdown of the things we need to think about in this space. So start off here, let's just think about what the green bar on the left-hand side is our potential MCDR. So imagine this can be the carbon locked up in a salt marsh, or the amount of carbon taken up in a particular volume of kelp or macroalgae, or even in phytoplankton. So this is our initial starting point to say, this is the potential we've got for marine carbon dioxide removal. But then you have to consider the baseline. So what is it that this is actually replacing? So here's two examples of what we need to think about. And the example on the left-hand side is mangrove restoration, and on the right-hand side, you have seaweed farms. So you can see before and after in both situations. So you can see, for example, the mangrove restoration, you can see that there's definitely an improvement, and you can see visually there's an increase in primary production and carbon uptake in that environment. But you have to recognize that even before, there's some level of carbon flux going on in this situation, and also fluxes of other greenhouse gases that you need to consider in terms of the background or the baseline. Similarly, on the right-hand side, you can see that with seaweed farms, the idea of these is to encourage growth of seaweed and the uptake of CO2. But these seaweed farms are replacing the natural uptake of carbon that would be going on through phytoplankton production. So once again, in terms of considering your net effect of these MCDR techniques, you have to consider what you're replacing. So you need to establish, even before deploying these MCDR techniques, you need to establish the baseline or background carbon uptake in these situations. Then, of course, you have to consider the operational side of things. So here I'm talking about the deployment of these techniques. I have a couple of pictures here of just examples of how you might do this through the release of lime or nutrients, through aeroplane deposition. And then, of course, there's the actual maintenance of these things. So there's a photo there on the right-hand side of a macroalgal farm. And so all of these have an energy cost and therefore a carbon cost in terms of finding the raw ingredients and then deploying the techniques. But an important extra consideration to consider in the operational side of things is the monitoring, reporting, and verification. So this is important for any outcome of the MCDR. The fact is that we need to have this not only in terms of the carbon markets that might be considering this approach, but also for national carbon inventories. And so any MRV, as it's called, will have once again an energy cost and therefore a carbon offset. And so once again, these operational costs will discount the total amount of MCDR that's happening through these techniques. And then we have the side effects. So here I'm talking about things like emissions of other greenhouse gases. The figure on the top right-hand side gives an estimate from the Rosenstretter paper of the emissions of of nitrous oxide and methane that might result from blue carbon, so from mangroves or seagrass beds, so that you actually might increase your uptake of carbon, but you'll actually also increase the emissions of these two greenhouse gases. So there's an increase in the kind of equivalent of CO2 production in these systems. And then there's leakage as well, where we have to consider that not all this carbon being fixed initially is going to actually make it to a point where it's going to be sequestered for long term, and there'll be other indirect effects. So once again, when you consider all of these different factors, you have to see that the potential that you have in these CDR is going to be diminished considerably when you take all of these things into account. So ultimately, you can see on the right-hand side of the figure, once we've accounted for those, your net MCDR or the additionality is much lower than the potential. So how do we establish this? Well, first of all, we need to understand the background variability. And so, as I mentioned, this has to be done pre-deployment, and it has to be account for seasonal and temporal variability. For the operational side of things, we have to do a full life cycle analysis of any technique. So that's from the actual startup of these operations, the way through to the end game of their end life. And then finally, for the side effects, we need to consider not only the other greenhouse gases, but a full analysis of where all the carbon is going in the system. For example, the release of dissolved organic carbon through these different techniques, and where that ultimately ends up. So we need some level of forensic carbon accounting to establish this additionality. Now, just quickly, I'm just going to run through very briefly about what other things we need to consider in terms of the other factors that affect, maybe the choice of MCDR that we go ahead with. So really here, I'm just comparing coastal blue carbon on the left to the open ocean MCDR techniques on the right. So factors we want to consider include, here I'm really talking about which one, the relative pros and cons of the coastal blue carbon versus the open ocean MCDR. And so there's much more regulation in the coastal zone. And so there's real advantages in terms of, building on existing regulations in coastal zones because governance is a very important issue for any technique that we deploy in the future. And similarly, because the coastal zone is so much more accessible than the open ocean zone, monitoring, research and verification, regulation and verification is much more accessible in the coastal zone. So there's a real advantage on the left-hand side here for the coastal blue carbon from these aspects. Then also there's the consideration of those side effects, and simply because of the scale of this techniques, probably the side effects will be less for coastal blue carbon rather than open ocean MCDR. And we're also well aware of the fact that coastal blue carbon has many co-benefits in terms of coastal protection. There's biodiversity advantages. There's more economic advantages of coastal blue carbon techniques than open ocean MCDR. And once again, all these different factors add up to the fact that gives greater social license for the deployment of things like remediation of mangroves and seagrasses and salt marshes. So looking at these factors, coastal blue carbon has advantages here, but then there's a couple of big things that we really need to consider. That if we want to have an impact on atmospheric CDR, we want to consider permanence. And assuming that we can get open ocean MCDR to be permanent, so that carbon's locked away in the deep ocean, this has a greater chance of greater permanence or sequestration of the carbon over a period of 100 years plus. And then the other factor we need to think about is scalability. If we're going to have a significant effect on atmospheric CO2, it's quite clear that it's only the open ocean that can really deliver this. Coastal blue carbon is great, but really the limitation on the availability of coastal real estate means that we can only make a small dent in the atmospheric carbon budget. And it's the open ocean where this is going to have an effect. But so the one other thing that, of course, that we need to consider is, what about ocean acidification? Just because we're actually lowering atmospheric carbon dioxide through these approaches, does it mean we're going to benefit ocean acidification? So let's have a look at this figure again a bit closer. And here I've just simply annotated each of these techniques with a yellow tick for a positive effect on ocean acidification and a red cross for a negative effect. And so moving from the left, if we consider the coastal techniques, they generally get a tick mark. We know that these are already being deployed to try and deal with acidification in places. There's some good examples, for example, from Tampa Bay in the US, where they're showing positive effects of seagrass meadows on raising pH. Then we move offshore and we have things like alkalinity addition. And once again, because you're adding carbonate and raising the alkalinity and the pH of the water, you're having a beneficial effect there in terms of ocean acidification. Then we move further offshore and we consider things like artificial upwelling. Well, the trouble with artificial upwelling is it's not only bringing nutrients to the surface to get phytoplankton to grow, but it's bringing extra carbon with it. So the net benefit might not be that great, and it might even be negative in terms of ocean acidification. Then the other two techniques which rely on using nutrients to stimulate phytoplankton growth revolve around the algal farms and the nutrient addition. And whereas these can have a beneficial effect in surface water, the sinking of these, both deliberately through algal farms or macroalgal farms, and also through the sinking of any fertilization event, will actually transfer the carbon to deep water, where it will have an acidification impact. And then finally, we have to consider the deliberate transfer of carbon to the ocean in the form of biomass and also in terms of liquid CO2, where once again, you're increasing the acidification issue in deep water. So once again, these are all things that we need to consider in the debate around whether and if we deploy marine carbon dioxide removal. So to summarize, lots of things to think about here around marine carbon dioxide removal, where we go from here. And particularly, I've just tried to raise 2 points here for you to consider: the implications for ocean acidification, and also that whatever we do, we really need to consider this important factor of additionality. Thanks for your attention.
Thank you, Cliff, for your presentation. Thank you for taking us through the net benefits, understanding marine carbon dioxide removal, the techniques and its implications in the context of ocean acidification. We welcome any questions that the audience may have. We do have a lot of time, so I'll leave the chat box open. Alexis mentioned that not a question, but she just wants to say how much she appreciates this presentation in your slides.
Thanks, Alexis.
Would anyone like to?
Can I ask a question?
Sure, go ahead.
In your expert opinion, what do you think is the best choice like a government should be making in terms of taking active action?
That is a very good question, and I think it's very dependent on which country it is and their situation. You know, this kind of very mini review that I've presented here, a very mini review, is partly in terms of trying to decide, or at least give some initial guidance on where New Zealand should go in this space. You know, New Zealand is very much like the Pacific Islands. You know, we're surrounded by the ocean. We have a very large EZ, and there is obviously considerable potential to use this for various things, including marine carbon dioxide removal. But of course, there's, once again, as I said at the start, there's a number of techniques, and I only probably just skim the surface of the techniques that are out there and being considered. And each of these have pros and cons. And so New Zealand, unlike the Pacific Islands, we're all relatively small nations with relatively limited budgets and opportunities here. And we really need an assessment approach where we consider all these factors to decide where we put our money and our resources and investment before we make any decisions about where we should go. So, Abby, that probably doesn't answer your question directly, but I think, you know, once again, I should point out that I'm not necessarily advocating for any of these, but I am advocating for a national assessment process to decide which and if we go down these routes.
I have a question, please, Azaria. Thank you, Cliff. Thank you very much, Cliff, for your presentation. That was very interesting and informative as well. I'm just curious to know what is the current status in the world of like what kind of effort that's been put through for this carbon removal so far, and has this been on the big scale or not yet, or something like this?
Good question. Yeah, I should have probably pointed out at the moment that when I actually, at the start of my presentation, that when I showed these different options that are being considered, you know, on the left-hand side of the finger, when we talk about the coastal blue carbon, we've already got techniques that are being put in place. As I said, you know, a lot of these are being used to good effect to deal with ocean acidification. But then when you move into the open ocean, many of these techniques have kind of been talked about. They're certainly appearing in models, and there are some small scale startup demonstrations of some of these techniques, but there's nothing that's actively being used at the moment. Certainly there is a lot of interest in some countries. I'm certainly aware of the fact that in the US and in Europe, for example, there is a lot of momentum in the space of funding research and development in some of these techniques, but as yet there's no actual even medium scale deployments of any of these. And I think that part of this comes from the fact is that not only there's a lot of questions about these things, but it goes back to what I said about the governance and legislation around these things, that this field is moving so fast that it's way ahead of the governance and legislation. And this really needs to be brought up to speed to actually regulate these activities.
Yes, thank you, Cliff. And there's also the upcoming COP 28 or meeting where these kind of things can go up to that level, and this could get more support from that kind of level. Thank you
Absolutely.
very much, Cliff.
Hello, I have a question.
Hello.
Yeah, just very, very briefly, perhaps not really a question. Thank you so much, Cliff, for that really interesting presentation. I enjoyed that. I'm wondering, there is a lot of talk about this being, about, the new part of the blue economy conversation that's happening, and particularly for the Pacific. I think more and more we're hearing this as being, this is part of our development for the blue economy. And I'm just kind of thinking about it in like, are there any good reasons for us to like think of it as a, yeah, I mean, if our leaders should come to us on this, and I feel you've answered some of it, but I wanted to take it down kind of in the blue economy kind of discussion. How if our leaders or members come to us, sort of, are there any good reasons for them to like try to look at this as being something to make money out of, or should it be a no? I don't know. I'm just like trying to get my head around the whole blue economy conversation that's going around as part of this.
Yeah, thanks, Katy. That's a good question, and actually it's quite an important question.
Can't hear you. Sorry.
Oh, can you not? Can you hear me now? Oh, can you hear me?
Yes.
Yes, you can hear me. Okay. Okay, so yeah, so good question, Katy. And obviously economics is an important aspect, because that's not something I touched on in my talk, and that will be a driver of governmental consideration of these things. I mean, clearly there is a market starting up for these kind of things. I mean, at the moment it's in most countries it's the level of a voluntary carbon market, but whether this actually is brought into there's large scale selling of carbon credits on the back of these things, where a nation or a region can make money on that is still to be developed. And I'm not too familiar with that field, but definitely I think that economic consideration is necessary. But once again, it comes down to, and maybe you're right, we should be thinking about this as another factor to be brought into those list of factors I presented, that there's the economic benefit or not of these techniques, in terms of, do you use a particular body of water in your EEZ for fisheries, or do you use it for carbon sequestration, or do you use it for tourism? And so it's a trade-off there in terms of the economic value of that. But then again, that has the trade-off of the environmental benefit that you get from that same body of water without doing anything to it, which of course that then comes into the environmental economics. And so that also has to be part of the conversation.
Thank you, Cliff.
Okay. Thank you everyone for your questions. We're reaching time, so I'll introduce our next speaker, Alleluia Taise, and she'll be speaking on the responses of kelp species to ocean acidification and light. Louis' PhD thesis focused on the effects of climate change and irradiance on the physiology of kelp species, and majority of her work has taken a multi-driver approach. She uses traditional physiological techniques to explore the role of inorganic carbon physiology and species ranges. Luya recently passed her PhD oral defense at Wellington, Victoria University of Wellington, and she now is a lecturer at the National University of Samoa. Over to you, Luya.
All right. Thank you, Azaria. Just trying to put my slideshow on. Apology for not showing my face on the screen. It's something technical issue going on with the laptop. But can you all hear me?
Yep, we can
Yeah.
hear you.
Okay. Thank you. Yes, so talofa and warm greetings from Samoa, everyone. So this presentation is based on one of my lab experiments that I conducted in Aotearoa, New Zealand, at Victoria University of Wellington as part of my thesis research. So for my thesis overall, I investigated the physiological responses of the green macroalgae Kalerba to environmental stresses of climate change, in particular, testing the responses to ocean acidification, ocean warming, marine heat waves, and light. So for today's talk, I'll share some of the key findings from one of my experiments on the combined effects of increased CO2 under ocean acidification and light. So Kalerba, along with all macroalgae, are important for the ecosystem services they provide. For example, they provide a three-dimensional habitat for many marine organisms. They contribute to coastal primary productivity. They are a food source for many marine organisms as well. But one of the reasons I'm interested in the responses of Kalerba is because Kalerba is edible in Samoa and in the Pacific. So this is the sea grape. And Kalerba is also an important fishery here because of its cultural, social, and potential economic values. So under ocean acidification, the responses of macroalgae may depend on physiological differences between species and the wider taxonomy groups. So there's substantial amounts of evidence to show that reduced pH as a result of increased hydrogen ions will negatively affect calcareous macroalgae because of reduced calcification rates. For non-calcareous macroalgae, they are suggested to either not respond to OA or be positively impacted by increased CO2. Anthropogenic sedimentation and pollution, they affect the light availability in coastal habitats, and any changes to light availability and quality can cause major changes to the physiological performance, geographical distribution, and depth zonation of macroalgae. So all macroalgae, they use CO2 for photosynthesis. However, some macroalgae, they could take up CO2 from the seawater through passive diffusion, while other macroalgae, they are able to take up another form of dissolved inorganic carbon, which is bicarbonate. And they do this through the active uptake process, which is energetically costly. So for those macroalgae that could use bicarbonate, we refer to them as CCM species because they have a CO2 concentrating mechanism that allows them to actively uptake bicarbonate. So if macroalgae, if they don't have a CCM, they are restricted to using CO2. And if they have a CCM, it means that they could take up bicarbonate actively. So we would assume that there wouldn't be as large a positive effects under elevated CO2 for CCM species. So in most species of macroalgae within the same genus, they either do one thing or the other. But Callirpa is interesting because it's one of the few genus where species could either have or do not have a CCM. And so here, like at Wellington in New Zealand, there are two most common species of Callirpa found there. One is Callirpa brownii, which does not possess a CCM. So we refer to this as the non-CCM species. And the other species, which is Callirpa geminata, and this one possesses a CCM. So we refer to this as the CCM species. So based on the differences in the dissolved inorganic carbon physiologies of macroalgae, especially for Callirpa, I set up to do an experiment, and I hypothesized that one, the non-CCM species will benefit from OA more than the CCM species, but the benefits for the non-CCM species would occur more frequently at higher light levels, because this is where the dissolved inorganic carbon limitation occurs for the non-CCM species. I also hypothesized in number 3 that the CCM species, Callirpa geminata, will only benefit from OA at lower light levels. And this is because at higher light levels, the CCM species will not benefit from OA because energy supplied to the CCM will not be limiting. So to test this hypothesis, I measure any possible benefits through increases in growth rates, photosynthetic rates, dissolved inorganic carbon uptake rates, and associated photophysiology. And I also use, especially for my hypothesis number 3, I use the stable carbon isotope values, or delta 13C values, to indicate any downregulation of the CCM, where the delta 13C values may become more negative. And I'll show a plot in the next few slides. So the aim of my study here is to use Callirpa as a model genus to investigate the interactive effects of OA and light on the green macroalgae Callirpa due to their unique carbon physiological differences, but also the differences in their light and depth habitat range. So I set out to do an experiment, and this was undertaken using a full factorial experimental design with 2 levels of pH corresponding to the present day PCO2 concentrations, and also an average value expected by the end of the 21st century under RCP 8.5. So the 2 pH levels were ambient 8.03 and future 7.63. I also had 4 environmentally relevant light levels, 20, 40, 60, and 80. And I did the experiment in a flow-through experimental setup. So I combined, or I crossed the 2 pH levels with the 4 light levels, and I ended up with 8 treatment combinations. And the pictures here show how it was physically set up in the lab. Again, this was translated into the physical setup in the lab, where we have 6 experimental tanks within a larger tank. And so the different colors here correspond to the different pH treatments. So red is for future low pH, and blue for current or ambient conditions, pH conditions. So here's some results. For the mean relative growth rates, we see that the mean relative growth rates of both species, they tended to increase with increasing light intensity, but the only significant effect of light on mean growth rates was found in the CCM species, Callirpa geminata, on the right here. In terms of photophysiology, I measured some metrics, including the maximum relative electron transport rates. And what I found is that the maximum RETR responses were not impacted by changes in CO2 and light, or the interaction of CO2 and light in the CCM species on the right here. So this reflects an insensitivity of the maximum relative electron transport rates metric to future high CO2 and low light seawater conditions in the CCM macroalgae. So this incongruence between maximum relative electron transport rates and growth responses of the CCM species, it indicates that photosynthetic rates were not stimulated in a sense, but rather energetic savings occurred under elevated CO2 at the two highest light levels. On the other hand, for the non-CCM species, Callirpa brownii, it exhibited pronounced upregulation of the maximum relative electron transport rates here at higher light levels under OA. So the trends in growth and photophysiology for the non-CCM species correlated. So I also measured the stable carbon isotope values. So here, what we're looking for is any reduction in the stable carbon isotope values or delta 13C values. And if the values become more negative, it's an indication of increased CO2 uptake, so going down the plot. So overall, from the results here, I see that the delta 13C values for both species were impacted differently, where the non-CCM species demonstrated more negative delta 13C values only at higher light intensities of 60 and 80 under OA. And this reflects a different photosynthetic pathway in these species compared to the CCM species, Callirpa geminata. So again, any reduction in the delta 13C values represents increased CO2 uptake, and this may translate into increased physiological performance and growth. And this was reflected in the general increased growth rates of both species under elevated CO2 treatments. But this increased growth pattern was insignificant for the non-CCM species. But also, this decrease in delta 13C values under OA could be also attributed to a decline in the bicarbonates uptake with increasing CO2 available. And this is because of the diffusive uptake of CO2 is energetically inexpensive relative to the active uptake of bicarbonate. And this is seen in the CCM species. And in contrast to the non-CCM species, it demonstrated a light-dependent change in the delta 13 carbon. So I also measured the dissolved inorganic carbon uptake. And here, the rates of dissolved inorganic carbon or DIC uptake during photosynthesis were significantly affected by light and pH, but not by their interaction in the non-CCM species. Here, I see that the highest light treatment, which is 80, it significantly increased the dissolved inorganic carbon use than all the other light treatments under future low pH treatments. So the dissolved inorganic carbon use responses in the CCM species were not impacted by pH, light, or their interaction. So while the DIC uptake and delta 13C responses may correlate at some point, they only imply that there is possible energetic savings or benefits under elevated CO2. However, to understand to what degree the algae rely on bicarbonate, I cannot infer that presently based on solely on the results that I'm showing here. So going back to my hypothesis, the results from this experiment support my first and second hypothesis that any benefits of increased CO2 for non-CCM species will be greater under higher light intensities due to the DIC limitations under high light. However, these findings slightly contradict my third hypothesis in this study, which I predicted that any benefit from OA in the CCM species will only be found at the lower light levels because of energy supplied to the CCM will be limiting here. What was observed was there was a general insensitivity of growth rates responses to elevated CO2 that were not reflecting in the photophysiology that exhibited more sensitive responses for the non-CCM species, for example. So this reflects that perhaps something else could be going on for these organisms. For example, increased hydrogen ions or low pH could also have negative impacts that are not portrayed in photophysiology and not just the CO2. So what some of the key findings in summary is that I found that the light had greater impacts than the ocean acidification or elevated CO2 on the growth and photophysiology in both Columbus species. So there were significant interactive effects that occurred between light and CO2. And that reflects that both light and seawater carbonate chemistry play a significant role in modulating photosynthetic and growth rates. And the results from this study emphasize the likelihood that elevated CO2 will act upon primary production differently, depending on the light and depth and DIC physiology of these macroalgae. And that's it. Thank you for your time.
Thank you, Luya. Thank you so much for your presentation and for sharing your findings of combined effects of ocean acidification and light on Columbus species. I can see Cliff has his hands up. I'll pass it over to you.
Thanks. Nice presentation, though. Very clear. Thank you. I've got two questions. One is a specific experimental question. One is a general question about the implications of your findings. So I just wanted to check, because I think I probably missed this in your first slide when you were showing your experimental setup. How was it that you adjusted the pH?
Oh, yes. Thank you, Cliff, for your question. So I used the CO2 gas, industrial grade CO2 bottle, and this was bubbled through to the experimental tanks. And in my experiment, I had a like the Apex tool, where it's like a cloud-based system where you can see what's happening in the tanks in terms of changes in the pH or temperature or things like that. And so you program this Apex system, for example, to respond or change when however you want it accordingly. So you have to program and put in some instruction into the program, into the software, and then the CO2 clicks on and off to turn on and off at some point. So, and also you can see what's happening in your tanks from wherever you are. You can just go on your phone and see how your pH is going, and if it goes over your target level, it will give you an alarm, and then you have to run to the lab and see if the CO2 is bubbling too much or it's not bubbling at all, and that's why the pH is going up or something like this. But yes, I used the CO2 gas.
Okay, that's interesting. I was just wondering if you'd considered the 13C signature in the CO2 you were using in the experiment.
Yes. Yeah. Yes. Yes, and I think I could also take that out as a potential cause for these responses. Okay. Is because we also tested for the delta 13C of the seawater in the experiment.
Okay, right. And my broader question was just about your last point there about your regime shift. So, I mean, I'm sure you're aware, having been here in Wellington, that New Zealand's currently experiencing an increase in exotic forms of caulerpa coming into New Zealand. Yeah. And I mean, do you have any feeling that what's driving this, and whether the carbon dioxide in the water is one of the factors?
Yeah, that's a good question, Cliff, and I think it's also one of the things that I can say I can discuss from the results of my thesis, because I also did some other experiments other than this one, looking at ocean acidification and light, but I also tested the responses of these organisms to ocean acidification on its own. And then I also tested these responses to just marine heat waves and warming. So, and then at the end, I combined all of these factors together. So it was really interesting that the responses were kind of variable and it depended on the condition. And so the question coming back to this, yes, that's right, Cliff. I saw some news about this annoying new caulerpa coming into New Zealand, and it could be these drivers of climate change, CO2 or temperature, because we also see that the warming and some marine episodes of like warming for a long time or like warming incidences that are not really familiar in this region. It's starting to come up again. So it could be these reasons. And the Kalerpa, I think the takeaway message for me as well is that these species, they are kind of hardy, and I think they could be happy at the conditions that under climate change.
Mhm. Okay, thank you.
Thank you.
Thank you. We do have 5 minutes left till the end of our session, maybe another 2 or 3 more questions before I'll hand it over to Yumeng to close up the session. I guess if no one has any questions for Luya, I'm gonna go ahead and hand it over to Yumeng. But before I do, I'd just like to thank everyone for taking their time out to be here, the audience, as well as the presenters for presenting today. Thank you.
Thank you very much, Azaria. And thank you everyone again for such a great session. So to wrap up this session, we would like to thank the organizers, the moderator, and the speakers for gathering here to give us such a great session. And we thank you, the audience, for joining us for OA Week 2023. Please do not forget to consult the website and to register for any other sessions that may interest you. If you would like to stay up to date with the GOA-ON community, consider signing up as a GOA-ON member on our website. And to discover more about ocean acidification research for sustainability, please scan this QR code. Thank you all, and see you next time.